A very good afternoon. My name is Vineet Upadhyay and I'm here to present my study on comparative genomics of plant serpins. Serpin is an acronym for serine protease inhibitor, and they constitute a superfamily of homologous protein with very different kind of functions spread across all life forms on Earth. And when they were studied extensively in vertebrates, they were found to have inhibitory as well as non-inhibitory functions. Why serpents are unique? Because they actually bind to the serine proteases covalently to the active site residues and block them irreversibly and modulate their functions. So Phytosome database released many new genomes of different species of plants, and we tried to catalog the serpins found if we can find and catalog the serpins in those new genomes. So the hallmark of serpin biology is a reactive center loop. The serine proteases cleave at P1P1 bond, and from about 20 amino acid long residues in the C terminal end constitute of the reactive center loop, which we can see the pattern which is highlighted here in the alignment of Brassicaceae family, the P17, 16, 15, 14, and 13, and with their corresponding residues which are conserved, we can see that we can highly identify if the protein which we have is whether a serpin or not. And since the reactive center loop is the first which binds to the serine protease, this actually determines the specificity of a serpin. So we divided our work into three parts. We collected the data using BLASTP searches and keeping the e-values below 10 to the power minus 3 in the plant genome databases, phytosome database. And we first did the characterization studies. We tried to predict the signal peptides, and then we searched for extracellular domain. We did the sequence analysis. We did alignment, phylogenetic studies, visualized in GeneDoc, and did try to determine the intron exon structure. And we tried to do structural analysis where we did homology modeling using ITASR web server. So we identified 318 serpins from 27 genomes present in the database, out of which 59 were either partial or did not have the necessary or the essential serpin domains, and some plant species did not had any serpins, which might be due to some human errors. But M. truncatula, Guttatus, and Domestica had the highest number of serpin hits, like 18, 6, and 9 respectively. We also tried to do um, distinguish serpins from other unrelated proteins. We tried to do some targeted signature based studies, and we tried to generate the gene structure of plant serpins. We actually found whether, although there might be a variability in the position, but most of the, like, out of 57 out of the 68 sequences were highly conserved at the intron 149C. And in Brassicaceae family, we found some variable regions. We found some high conservation near the reactive center loop at position 308 and position 333. And 13 of the 50 sequences analyzed had the insertion in this region. And we also tried to do some studies using to find if there are any extended domains using PFAM. On the right hand side of the figure, the yellow and blue mark are the extended C terminal region and extended N terminal region found in some serpines. And also you can see on the left hand side there we can see that the locus expansion has been done differently, maybe due to evolutionary due to during evolution they might have been doing some different evolutionary functions, but since all they still have a conserved domain at intron 149C. Also, we tried to do build some genome structures and try to annotate them accordingly. Thank you to DBT and IRB for funding my project, and special thanks to Dr. Abhishek Kumar for giving me the opportunity. Thank you. All questions are welcomed.